Hello everyone, this is your educator Priyanka Thakur teaching you life sciences and welcome you all on our YouTube channel Gurmantra Shiksha Ka Granth. So I have started with microbiology and today I'm going to teach you about nucleoid and plasmid. Okay, I'm not going to cover plasmid from the biotechnology perspective, but rather than definitely I'll be covering it from microbiome perspective. I'll be telling you about different types of plasmids, not like bag or yak plasmid, okay, but the other type of plasmids like F plasmid, conjugative plasmid, transmissible plasmid, etc. And you will also come to know about copy number, partitioning, and we will also discuss about nucleoid. So let's start with our today's topic, which is about nucleoid and plasmid. So first of all, I would like to tell you about nucleoid. I know you already know a lot of things about nucleoid, fine. So today we are mainly going to discuss about nucleoid, fine. Okay, nucleoid. So nucleoid is what? Nucleoid is what? Like, is a region of bacterial cell that will contain a bacterial chromosome. So we can say nucleoid contain bacterial chromosome. How many bacterial chromosome are there? In Normally in eukaryotic cell there are a lot of number of many number of, like, there are many uh, chromosome, but in bacteria there is it is the nucleoid represent only one chromosome. That is, does it have a limiting membrane? It have no limiting membrane. Okay. Does eukaryotic chromosome have any limiting membrane? Yes, they have, but no limiting membrane is there in nucleoid. Okay, and it does not contain any basic histone, no histone proteins. We can say it is not associated with histone proteins. Rather than histone proteins, it is associated with polyamines. Okay, it is associated with what? Polyamines. Fine. Other than these polyamines, there are some other proteins which are known as nucleoid associated proteins. Other than polyamines, there are no histone protein, but other than polyamines, there are some nucleoid associated proteins, and they are named as HU. Have you heard about HU? HU. What does HU indicate? It indicate heat unstable. Then comes your H and S, means histone like, you can uh, call it histone like nucleotide structuring. Fine. Okay. Then next comes your IHF. And what is IHF? IHF says integration host factor. Integration host factor. Fine. You, if you want, you can watch the video on higher speed also. And then come SMC, structural maintenance chromosome. You must have heard about all these things already, all these proteins already. May not be, but you must know about SMC, structural maintenance chromosome. What is the function of structural maintenance chromosome? You already have covered it in some of your topic, for sure, I know. Okay. And these are the main nucleoid associated proteins. Out of these proteins, our SMC is involved in chromosome segregation. Remember, it is involved in chromosome segregation. The main function of SMC is chromosome segregation. And other one, like your HNS, histone like nucleotide structure, is mainly involved in chromosome, or I can say DNA condensation. And other HU and your IHF, they are involved in DNA banding. Right? You understand the function of all these genes? Fine. So IHF, now told you about IHF is an example of protein that will be involved in banding of DNA. Fine. All these proteins helps in integration, inversion, recombination events by banding the DNA. Okay, most of the bacteria, if I'm talking about E. coli, okay, how many bacteria, how many chromosomes are there in E. coli? E. coli will have a single circular DNA molecule, okay, single circular DNA molecule. All the Does all the bacteria have single circular DNA molecule, or and is there any other bacteria which may have two bacterial chromosomes? So, the recent studies indicate that some bacteria may contain more than one chromosome. It may be linear or circular in nature. If I'm talking about Vibrio cholerae, you know the Vibrio cholerae may cause cholera. This bacteria contains two circular DNA. Okay, two circular DNA are present in this Vibrio cholerae. Now two circular DNA are there, matlab, it means what are they going to do? One of them is involved in metabolism in and virulence. So one will be involved in metabolism and virulence, and other will be doing what? The other one will contain some other essential genes. Okay, the other will be having other essential genes. Fine. So a Vibrio have how many chromosomes? Two chromosomes. Then comes your Borrelia. Okay, Borrelia, I would say. Borrelia. Fine. Now Borrelia, double R will be there. Okay, Borrelia. Borrelia. I'm sorry. Borrelia, this is a bacterium that causes Lyme disease. You do not forget it. That Borrelia may cause Lyme disease, and this bacteria contains 11 copies of DNA. 11 copies of DNA, and it will be single linear DNA. Fine. Remember, E. coli have single circular DNA. Vibrio have two circular DNA. Borrelia that causes the Lyme disease, it it contains 11 copies of DNA, and those also linear. Okay, so DNA molecule of archaeans shares many similarities with the eukaryotes. Archaeans are the only group of prokaryotes that use eukaryote like histones. Fine, they can condense their molecules like DNA molecule of archaea. It can also be negatively supercoiled, positively supercoiled, or even without any supercoiling. So you know about bacillus. How many chromosomes are there in bacillus? One circular, just like that of your E. coli. You know about tumefaciens, agrobacterium tumefaciens, right? Yes. And how many chromosome does it have? Okay. So agrobacterium tumefaciens have one linear chromosome. Fine. And you know about E. coli for sure. E. coli have how many? One circular. E. coli, the genome of E. coli is known as K12. Okay, genome of E. coli is known as K12. Remember this thing. The genome of E. coli is known as K12. Fine. Now let's talk about what is the difference between what and what. Prokaryotic DNA. I'm not going to write down the differences. I will be asking you what is the difference between prokaryotic DNA and eukaryotic DNA. Okay, prokaryotic DNA and eukaryotic DNA. So you are going to tell me the difference. First thing, tell me. Mostly prokaryotic DNA are circular. Okay, they have single circular chromosome. Most of all the time, they have single circular chromosomes. While eukaryotes, they have many linear or circular. Tell me, linear or circular, they have many chromosome, they have more number of chromosome as compared to your prokaryotic DNA. Actually, the prokaryotic DNA represent is equivalent to the one single chromosome of eukaryotic cell. That's why it is considered as chromosome. Otherwise it is not even a complete chromosome. Exactly, these are more generally haploid in nature, and these are diploid in nature. Fine. And the prokaryotic cells may contain many plasmids, okay, while rarely present. Plasmids are rarely present in eukaryotes. You cannot say, excuse me, okay, you cannot say that there are so many plasmids in eukaryotes. Rarely, yes, rarely plasmids are there. Rarely plasmids will be present. Have you ever heard about plasmid being present in the eukaryotic cells? So we cannot be so assured about there are so many plasmids that could be present in the any eukaryotic cells, or many eukaryotic cells are there in which plasmids could be present. So rarely plasmid will be present in eukaryotic cell. I will be telling you in the end which cell is containing plasmid and what is the name of that plasmid. So prokaryotic chromosome are present in condensed form in the nucleoid, and as we know, there are no histone proteins complexed with the DNA, but histone proteins will be there complexed with the DNA. Fine. Next comes our size. The DNA of prokaryote is much smaller as compared to eukaryotic DNA. So we can see that eukaryotic DNA is comparatively larger than prokaryotic DNA. Fine. If we talk about K12, I'm giving you an example. If we talk about K12, which is the DNA of E. coli, chromosome of E. coli, it is roughly equivalent to 4.6 MB. And if we talk about yeast, fine. So yeast is having genome of approximately 12 MB. You understand? So prokaryotic, they have a very less non-coding region. Rarely they have any non-coding region, like introns. And here you will see many introns, like many a lot of non-coding region will be there. Okay, we need to splice the eukaryotic mRNA in order to form a mature mRNA. The first mRNA formed is heterogeneous mRNA. Fine. Now let's talk about plasmids now. We covered our what? We have covered our nucleoid. We'll be talking about plasmids. Ma'am, you told us only some basic things about nucleoid. Definitely, these were the basic things, but there were also some things that were quite important about it. So, plasmids, the term was coined by Lederberg. Okay, Lederberg was the person who coined this term. Okay, are they self-replicating? Definitely, plasmids are self-replicating, and they are what? They are extra chromosomal segment of DNA. They are extra chromosomal segment of DNA. So we can say they are extra chromosomal genetic material in bacteria. Well, other than bacteria, other than main chromosome, this is a segment which is present and it is also genetic material. So extra chromosomal genetic material in bacteria. Okay, fine. Okay, is it a replicon? It acts as a replicon, so it can act as a replicon. Are there any RNA plasmid or not, or only DNA plasmids are there? They are mostly DNA plasmids, but rarely they can be RNA plasmids. RNA plasmids can also be present in some like plants, fungi, and other viruses also, even in some animals. So we can say RNA plasmid can be present in some viruses, like in plants mainly, fungi, and in some animals also. Fine. So they are usually smaller than the bacterial chromosome. They are not larger than bacterial chromosome. They are smaller than bacterial chromosome. They are extra chromosomal segment and they are smaller than bacterial chromosome, not larger than bacterial chromosome. Fine. They are hardly 5 kbp kilobase pairs to more than never only 100 kilobase pairs. Though plasmid are as large as 2 MB, they can attain the size of 2 mbp. Okay, in some bacteria, they encode the trait which are not exactly essential for bacterial viability, and they can replicate independently of the chromosome. Mostly these are circular. Most of the plasmids are circular, negatively supercoiled. Okay, most of the plasmids are circular and negatively supercoiled. They may be double-stranded DNA molecules, but linear plasmids are are also seen. Okay, but linear plasmids are also seen in which bacteria, like streptomyces. Okay, streptomyces contain your linear plasmid, right? Now let's talk about types of plasmid. We are going to talk about types of plasmid. What are the types of plasmids? So plasmid is mainly of following types, in which the first one is your conjugative, and then comes your transmissible, mobilizable, I would say mobilizable, and then comes your self-transmissible type of plasmid. Now we will discuss about these plasmids. First of all, let's talk about conjugative one. These are large plasmids. They carry genes for the formation of specific donor recipient pairs. It means they have genes for the synthesis of pili. So remember mainly about these conjugative plasmid, they have genes. They have genes. They have genes for the synthesis of what? Pili. Very important. Conjugative plasmids, they have the genes for the synthesis of pili. Then come non-mobilizable plasmid. So this non-mobilizable plasmid is not self-transmissible. It cannot it cannot be move itself. It cannot move itself, so it is not self transmissible. If it is not self transmissible, it means it does not have TRA genes. So it lacks TRA genes. TRA genes are not present in the plasmid, who are not self transmissible, but they can be transferred with the help of other transmissible DNA. They can be transferred with the help of, they can be transferred with the help of other self transmissible DNA. Fine, remember this thing. And for this they need a gene. For this they need a gene. And the gene is known as, like, they are not having gene mainly, but they have a site. They have ori T site. Remember this site, the DNA which does not have TRA genes, they have ori T site, and such DNA are known as mobilizable. Like, these are the plasmid which are not self-transmissible because they do not have TRA genes, but they can be transferred by self-transmissible plasmid, and such a plasmid called mobilizable plasmid, and the process by which they are being transferred is known as mobilization. So this transmission lies on the product of TRA genes, which, and these TRA genes product are specified by other self-transmissible plasmid to achieve the transfer, and mobilization plasmid must contain an ori site. Fine. And third one is our self transmissible. Okay, so these are the these are these such plasmid, they will have TRA genes. Definitely they will have TRA genes, and they encode all the protein that is required for transfer between donor and recipient. It is both, like, it is both conjugative and mobilizable. It is both conjugative and mobilizable. Okay, if a plasmid that lack genes necessary for effective contact and DNA transfer is termed non-transmissible plasmid. Most naturally occurring plasmid are either self-transmissible or either mobilizable. Okay, now let me give you an example. F plasmid, which is an example of self-transmissible plasmid. Okay, so F plasmid is what? F plasmid, you must have heard about F plasmid. It is a self-transmissible plasmid, right? Okay, so will it have any TRA genes? Surely it will have TRA genes, because only those plasmid who have TRA genes could be transmissible plasmids, right? If a cell is having, this is a cell, okay, it is having its own DNA. Along with it, it have this plasmid. Okay, so this is the nucleoid. This is your F plasmid, and this is your F positive plasmid. Okay, this cell is known as F positive. If here is another cell, okay, which is not having any plasmid, F plasmid, this is only nucleoid, so this is known as your F negative. Fine. And if this plasmid gets incorporated into the nucleoid, so what will happen? If this plasmid is incorporated into the nucleoid, okay, this is the plasmid, it has been incorporated into the nucleoid. Now this is known as apisome condition, but cell is now known as HFR. HFR, high frequency recombination. Okay, so integration involves homologous recombination between two covalently related circular DNA molecule, forming one circular molecule containing both of original DNA structure. It is thought that insertion sequence present in F plasmid and those in the host chromosome serve as region of homology for insertional events. Okay, so is this is F plasmid clear to you now? Okay, let's talk about R plasmid now. R plasmid is also known as your resistance plasmid. Okay, our plasmid is also known as resistance plasmid. Definitely it is going to provide resistance to bacteria against antibiotic. So it gives resistance against antibiotics, right? Fine. Some R plasmid have only one single antibiotic resistant gene, whereas other have many. More R plasmids are mobilizable plasmid. They are mostly mobilizable plasmids. So remember this thing about R plasmids, and they can have resistance for one gene. They can have resistance for one gene or for many genes. Okay, so remember this thing also. And next come your coal plasmid. Coal plasmid is also known as your colchicine, not colchicine, colcinogenic plasmid, right? Okay. Now, what is the function of this colcinogenic plasmid? Uh, Actually, col plasmid, it synthesizes the protein bacteriocin. It synthesizes the protein bacteriocin, and this bacteriocin can prevent the growth of bacteria. Okay, and it will only be able to prevent the growth of bacteria who are susceptible to it. Fine. And most of the bacteria who are susceptible to this bacteriocin, they do not contain coal plasmid. Okay, they do not contain coal plasmid. That's why they are mostly susceptible to bacteriocin. Otherwise, if the plasmid which is having bacteriocin, coal plasmid, if they are having coal plasmid, they will be able to synthesize bacteriocin and they won't be susceptible to this bacteriocin. In E. coli, this bacteriocin is called as colicin. Fine, fine. There are a number of colicin, like they will be colicin A, B, etc., and each having a particular mode of inhibition of sensitive cells. Okay, so what are the types of plasmid and their gene function? Like I told you about resistance. Okay, this is a plasmid like type. Okay, type of plasmid. You will also come to know properties of plasmid. Fine, types of plasmid. So this one is resistance. Okay, and antibiotic. What kind of resistance? Antibiotic resistance. Virulence, if plasmid is virulent, its property will be like its function will be pathogenic. Okay, if it is a killer, so what will be the function? If it is a killer, it will synthesize toxins and kill other bacteria. If it is fertility factor, then it will be helping in conjugation. Okay, if it is degradative, then what it will synthesize enzymes that will cause metabolism of unusual molecules. Fine. Okay, now let's talk about properties of plasmids. Very important one. Properties of plasmids. Now, what are the properties of plasmid? First of all, we will talk about replication. First one is replication. I already told you that they are self-replicating DNA molecules, extra chromosomal segment. So typical plasmids are circular, double-stranded. You know that, and they have two mechanism. One is just like that of bacteria. Okay, normal replication that takes place. So one is just like that of bacteria, and other one is your you have heard about it. Yes, it's rolling circle replication. You know about rolling circle replication. What happens during the rolling circle replication? So during rolling circle replication, it will start by nicking at the origin of replication and unrolling one strand and using the other still circular strand as template for DNA synthesis. Some plasmids, such as your F plasmid of E. coli, know about F plasmid. F plasmid, they can transfer themselves to one bacteria from one bacterium to another. So F plasmid have two origin of replication. They will have two origin of replication. One is the vegetative one. Okay, so that one is known as ori V, and other one is for transfer, that is known as ori T. Fine. So F plasmid who can be transferred, they have two center of origin. One, it will be the bidirectional replication will occur. So bidirectional replication is also known as vegetative replication, and it will start from ori V while the transfer starts from ori T. So all the plasmid must have a vegetative origin, since they all replicate to survive, but only those plasmid that can transfer themselves will have this origin of like transfer origin or IT. Okay, now let's talk about second property, which is host range. Now, what do you know about host range? When you say host range, what is host range? So, mostly plasmid can exist in a limited number of closely related genera. So, they either have narrow host range or they have broad host range. When will they have narrow host range and when will they have broad host range? See, if they are present, they are like they can exist only a limited number of closely related genera, then they fall into narrow host range. But if the bacteria can be transferred and maintained in bacteria of a large number of species, okay, they have no limit, okay, then they are known as broad host range bacteria, right? Narrow host range, broad host range. If bacteria can survive only in if plasmid can survive only a limited number of species of bacteria, then it falls into narrow host range. If it can be maintained and it can survive into many species of bacteria, then it is known as broad host range bacteria plasmid. Okay, next property is your copy number. When you say copy number, what you understand by copy number of bacteria now? So copy number, it is average number of molecules of plasmid per bacterial cell. Okay, or you can say it is the number of copies of plasmid. You can say number of copies of plasmids in a cell immediately after cell division. Okay, immediately after cell division, or other than that you can also say it is the average number of molecules of a given plasmid. If it is a number, average number of molecules, whose molecules, plasmids molecules, uh, per bacterial cell, per bacterial cell. Fine. So now copy number, it can be high or low. Copy number can be high or low. When there are only 1 to 2 copies of plasmid present, this is what we call low copy number. When there are about 100 and 10 to 100 copy number present, this is what we call high copy number. Okay. So the initiation and plasmid replication may be controlled by regulating the amount of available primers for the initiation of DNA replication. Other one is regulating the amount of essential replication proteins, or regulating the functions of essential replication proteins. So three measure, there will be like three measures. We are talking about copy number, and copy number will be obtained by replication. So three major mechanisms are used to control the initiation of plasmid. First one is your regulation of antisense RNA. Okay, so first one will be regulation of antisense RNA, then regulation by ctRNA. What is ctRNA? It is counter transcribed RNA, counted transcribed RNA. Okay, and other one is third one is regulation by binding of replication protein to what? Replication proteins to sites which are called introns. So these are the three major mechanisms that are used to control the initiation of plasmid replication. Three major steps that are used to control the plasmid replication. Okay, now let me tell you. Oh, okay, I have forgot to tell you about plasmid incompatibility. Okay, let me tell you about plasmid incompatibility. Fine, this is plasmid incompatibility, right? Now what is plasmid incompatibility? This is your next property, fourth property. What is plasmid incompatibility? So incompatibility is when two closely related plasmid, they coexist in the same cell. This is cell containing incompatible plasmid A and B will give rise eventually to descendant containing either A or B, but not both. Two plasmid that cannot coexist. Remember, what is plasmid incompatibility? When two plasmid cannot coexist in the same cell, same bacterial cell, this is what we call incompatible group. Okay, this is what we call incompatibility, and the two plasmid are incompatible if they belong to same incompatible group. If there is a plasmid and B plasmid, if we are trying to put them in the same cell, they won't come in the same cell. They will be in different cell. Okay, like this is the same bacteria, pseudomonas. Okay, pseudomonas, they are denying to be expressed in the same cell. Okay, then they fall into same incompatibility group. They fall into same incompatibility group. And the fifth property I will be telling you about is your partitioning. Partitioning is used to ensure the equal partition of chromosome into two daughter cells. If there are high number of copies of plasmid, then surely each daughter cell will get one copy of plasmid. But if the count is low, then maybe some of cells will be there who won't be able to get the copy of plasmid. That's why partitioning takes place. Partitioning is a your regulated mechanism which mostly occur in those cells who are having a low copy number of plasmid, and it is regulated by genes known as Part genes. Part genes regulate this partitioning process. So partitioning is regulated by part genes which are present on plasmid, and part system will consist of cis-acting centromere site which are known as pars S. Okay, cis-acting, they will have cis-acting centromere sites which are known as par S. Okay, and there will be trans-acting proteins also which are par A and B and are encoded by par A and B genes. Okay, these are par A and B genes. They are trans-acting, they are cis-acting, and these genes are responsible for partitioning. Okay, so now you know the function of bacteria, and I have already told you the function of bacteria. If you are talking about a cryptic plasmid, so the plasmid which is having no phenotypic properties is known as cryptic plasmid. When you talk about antibiotic resistant E. coli, Salmonella, Staphylococcus, they are antibiotic resistant. When you talk about pilus, E. coli, Pseudomonas, they have pilus. Insect toxin synthesis, Bacillus thuringiensis, you have heard about. Gas vacuole, Halobacterium. Oil degradation, Pseudomonas unona. Oil degradation, okay, whenever ship are there on the shores of sea, there are spills of oil, and these spills of oil are dissolved by using Pseudomonas. If insect toxin synthesis, Bacillus thuringiensis. Plant hormone synthesis, Pseudomonas. Antibiotic synthesis, Streptomyces. Okay, other than this, let me tell you one thing again. Okay, I forgot to tell you, and I'm not having for the sheet. Oh, okay. uh, are plasmid present in bacteria, eukaryotes also? So plasmids are also there in yeast, and the plasmid in yeast is known as 2 mu circle plasmid, remember. Okay, which is about your 6.3 kb circular extra chromosomal segment found in the nucleus of yeast, Saccharomyces. Okay, so tumor circle is coated with nucleosome, and replication is initiated by host replication enzyme once per cell. So that's all for today. Okay, so if you want to join an academy for CSIR net preparation, so here is the detail of paid classes on your screens. You can check out for how many months you need to pay how much. You can get 10% off from this particular fee also by using the referral code Neha, which is the referral code of Neha Tanisha ma'am. Go there and follow her on an academy. Along with this, do not forget to subscribe to our channel Gurmantra and join us wherever you want on a single click by looking down the description box. Check out the description box. You will get links to our different platforms on a single click. You will be there with us. I hope you got to know about a lot of things. If not, then definitely I'll make sure that in next time I'll be providing you with much more